Welcome to the Axial Podcast. Axial is an early-stage investment firm based in San Francisco. We partner with great founders and inventors investing in early-stage life science companies often when they are no more than an idea. Axial is fanatical about helping the rare inventor who is compelled to build their own enduring business.
Okay, I think we're record, Hans. Well, it was great to chat up last 10 minutes or so about kind of just like debriefing and everything. But how are you? Thanks for doing this.
I'm doing awesome, as always, I like to say.
Well, I'm really excited to talk because you've been at MIT for like a relatively short period of time and been hyper productive. And so I just wanted to have this conversation and discuss your research around machine learning and molecular dynamics, and then kind of what else is on the horizon. But maybe to like backtrack it and just understand your research a little better, maybe we can talk about your story briefly. And you kind of started your career off at Munich, and you've been at MIT for like 7 months, or maybe 2 months, but maybe all in all 7 months. How did you get involved in like science, and where did your journey begin?
So how I got involved into science and where my science journey began, that was basically so I started my master's at Technical University of Munich in like 2019, October 2019. And then in October 2020, I started my first like research project where we need to do this interdisciplinary project at Tommi as part of our courses. And then there I just reached out to so most people I think do it at some company, but I reached out to some lab at the university and asked them if they want to supervise me for a project. And I proposed, I read a few papers, and I was watching Yannick Kilcher's videos, in case you know him, and had some ideas on projects related to proteins. And yeah, they were a bioinformatics lab. I proposed a few projects to them, and then in the end we ended up working on subcellular localization prediction, which was absolutely unrelated to the things I proposed, but it was interesting to me. So yeah, that was my first research project, and I guess I absolutely loved it. And then decided that I want to do a PhD.
Cool.
And yeah, then I just finished that project in around, I guess, February. And then in March, I had to write my master thesis, or I was looking for a project for a master thesis. And yeah, for that, I actually in 2020, I went to my first conference. I just attended the online Europe's 2020. And yeah, from that, there I met a bunch of people and asked them if they want to supervise my master thesis on this or that topic.
Cool.
And they agreed. And then I wrote my master thesis with them. And, that was the 3D InfoMax paper, which is now also published at ICML. And yeah, after my master thesis, during my master thesis actually, so that was now the summer 2021, I was reading all of these geometry processing papers, and one of those was a paper by Octavian Garnier, who we got some ideas. While we were talking, I implemented those ideas and sent him the results, and he seemed to like that. And yeah, Octavian was here at MIT when we were talking about his papers, or about his paper. And yeah, then I mean, he seemed to like it that I just implemented some of those ideas, sent him the results. When I then asked him for an internship in October, he said yes. And at the same time, yeah, then in October we started working on EquiBind, this other paper that I guess we'll be talking about. And yeah, then I wrote my applications to a bunch of universities. And in February, I was accepted to a bunch of universities and then decided that I would join Tommi, Jaakkola, and Regina Barzilay. Yeah, at MIT. And that's, I guess, where I am right now. Because I started my PhD a little bit earlier, actually, than yeah, usually people start in fall, right? But Tommi offered me to start in June already, and that's what I went for.
Cool. I think it'd be great to dig in deep into InfoMax as a way to kind of predict kind of small molecule features, and using like a clever way to pre-train models, and then EquiBind as a way to kind of predict ligand binding to different targets, and using a clever sampling, which I don't understand completely even after reading the paper. Yeah, I mean, I can't. Also, the labs you're working in, I can't pronounce your last names, so I'm gonna have to rely on you for that. It really, we'll post the labs in the description, but their last names have like 5 syllables each, so it just I'm not gonna try even. But what was the logic? You know, you're trained as a computer scientist engineer, and you're making it. You're in Munich, and you're collaborating with people. What was the kind of spark to point your kind of expertise towards problems involved in chemistry, biology, life sciences overall? What was that kind of spark that kind of got you working on these problems?
So there are basically two main reasons. One is that I just think these areas offer very interesting types of data and are very methodologically rich, and you if you work on problems in these areas, you can come up, or you have to come up with super interesting algorithms or approaches to use to solve the problems, and you can bring ideas from physics, where a lot of statistics methods also are coming from. You can bring ideas from physics into the machine learning world and build interesting methods, and just make machine learning methods themselves, general machine learning methods themselves, better. And yeah, you also have data types like graphs or 3D objects that you're dealing with, instead of just the usual images, for example. And maybe then the priors, or the prior knowledge of how these objects interact and things like that, you can build into your models, and that is I think very interesting. But the other the second big reason is that I want my work to be impactful, or like this might sound what do you call it, like saying it from a high horse maybe, but yeah, I want the work that I do to be helpful for humanity.
Cool.
Or yes.
Cool.
What Joshua calls it, AI for good, I think. Yes, I want to do AI for good. So
okay, cool. Okay, you know, better than AI for bad. So yeah,
that's maybe not only AI, right? It is important to me that the work I do, like my professional life or my professional work, has a positive impact on humanity, or maybe also as positive as I can possibly do it. There's also this 80,000 hours concept, that right, you spend 80,000 hours in your life, working, like on average, maybe, I'm not sure how they came up with this number, but those 80,000 hours, you should, or maybe it is interesting to spend them as efficiently as possible for improving or for bringing humanity forward.
Absolutely. And so on that concept of spending time, how did you, on the machine learning side, I'm not an expert at all, I just kind of know a little bit and have friends, how did you determine to spend your time on graphs and graph methods and that kind of area of machine learning? Is there some sort of advantage there in terms of its relevance for chemistry biology, or is kind of graph-like methods, is there a lot of kind of new opportunities that are underexplored?
That's what I think. I am of the opinion that we should build as much prior knowledge as possible into our machine learning models if we want to solve a particular task or a particular problem. If you want to build AGI, then sure, maybe, or if you want to build a general reasoning robot, then maybe building in all prior knowledge that you have, or as much prior knowledge as you can, is not the optimum. But I think if you want to solve a particular problem that has that is useful to in the real world, then we want to build in as much prior knowledge, reduce the space of functions that we have to model, and bake in symmetries such that we can generalize to all symmetric cases. And using graphs is one way to do that, right? Or if we have some data that is that has a specific graph structure, we can process the data ignoring that graph structure, or we can use the graph structure. And then there's the question of how to use the graph structure and that prior knowledge in the best possible way. And there you then get into interesting graph neural networks and message passing neural networks and different approaches. And I just think this is then also a very interesting data type in itself, which makes it interesting to come up with new methods to process graphs better.
Cool. Okay. That's pretty interesting, actually. Okay, so graphs allow you to maybe model prior knowledge more efficiently, and especially in biology and chemistry, that is actually really useful because we still a lot of the knowledge is implicit. Maybe we can use InfoMax as a case study of kind of that kind of using the power of graphs. And so maybe you can talk. InfoMax is like your first major preprint, in paper piece of research, or maybe second after the first project. Can you talk about that story of InfoMax and how that project got started? Maybe to connect it to graphs and using prior knowledge. Maybe that was kind of a theme of the project, and then maybe talk about some of the discoveries, and we can just go from there.
So that project we started in like March 2021, right? And that there I had my great supervisor, my great advisors, Dominique Pierini, Prudenzi Tussaud, Gabriele Corso, who's now my colleague, because I joined him here at MIT, and Pietro Lio, also Stefan Günnemann and Tom. And basically I reached out to Gabriele and Dominique I read their very good, very interesting principal neighborhood aggregation paper, and also talked about their directional graph network. So I knew that they also were interested in graphs or working on graphs. And my position was basically I'm interested in working on graphs and applications to molecules, and that's then how we came we brainstormed more or less, and we knew that we wanted to do some bring the power of pre-training to the world of molecules, right? Because we have very, or often in the molecule world, we have very little data. Yeah, a small amount of data. But the pre-training approaches that are out there are often not very principled, because they we just mask out some atoms or something like that. And while if you just mask out an atom and then you try to predict it with similar to masked language modeling, right? Then yeah, you can probably get right almost, I don't know, 80% of the time by just predicting that the atom that you masked out is a carbon. So yeah, we just think that the pre-training approaches that were out there for molecules, they weren't very helpful. And yeah, what is it? And then the question was, what is it that we could have as a pre-training task that would help the model make better predictions and help reason about the molecule? Well, if the model knows about 3D structure, then it could plausibly make better predictions about those molecules for tasks where the 3D structure is important. So we devised this pre-training task in which the model is trained to reason about the 3D structure of small molecules, and then generates this latent 3D information, which it can then use to make better predictions for molecular properties. And there we have 3D InfoMax.
So what was the kind of the why now, in terms of there's people kind of knew this is some of a problem where there's a lack of sparsity of data, 3D data on small molecules or certain molecules because it's expensive to generate at scale, and then but there's a need to have better models. What was the kind of the breakthrough to train models to implicitly predict 3D structure and then improve predictive power? And then maybe you can talk about the things you predicted, because some of the properties were very a little complicated in terms of various quantum properties. Maybe you can discuss kind of like the readouts and what actually improved for the stuff you kind of were able to test on.
Yes. So in our evaluations, we found indeed that for quantum mechanical tasks, the improvements were quite significant, sometimes there were 22% improvements on average on the QM9 datasets, for example, for predicting its properties if we did our pre-training. Yeah, and that, I would say, makes sense for quantum properties, right? Because it's an intrinsic property of a small molecule. And if you have information about the 3D structure, then we've also seen with models like DimeNet, also from Stefan Günnemann's group, or spherical message passing, or GemNet, which use the 3D structure of small molecules in a smart way. There we've seen, but they need the ground truth 3D structure as input, of course, which our method does not. There we've seen also large improvements on these quantum mechanical tasks. And I think if you have, again, intrinsic property of a molecule, then predicting it while only having or additionally having the 3D structure available will help. But if you now just have a small molecule and you try to predict its toxicity without having any of the structures that it binds to to have its toxic effect. If we only look at the small molecule, and then if we additionally have the 3D structure, yeah, it might not help so much if we don't have the 3D structure of the molecule that it interacts with. And that is also what we found with 3D InfoMax, where we then didn't see improvements with the 3D pre-training when evaluating on tasks like blood-brain barrier penetration or the TOX21 challenge, or well, actually, then there were tasks like solubility prediction again, or the lipophilicity, where the 3D pre-training helped, right? Because these are intrinsic properties of the small molecule. Yeah.
I think the paper was a really elegant way to convey the power of pre-training in models, and then and at least for intrinsic properties of these small molecules. And what kind of from that we published the research and you got accepted ICML, so congrats on that. Or and so what did that unveil? Did InfoMax, did you often great research role unveil new problems or new things to work on? Did that project set up EquiBind, or was EquiBind totally separate? Or what did you learn from InfoMax that then like set you up for the research you did later on?
Oh, well, what did I learn from
the project? I'm sure you had all these different
but it's
challenges and whatnot, and yeah.
It is just so much that I'm not sure what I should pick out now. I definitely learned a ton in that project, from writing a paper to well, of course I've written another paper before, but yeah, there I really got into like the conference style of writing a paper, or the conference paper writing style, right? Because there was a conference submission, but I also learned a lot about working together with experts from chemistry and industry partners. I learned a lot about interacting or reaching out to people and discussing papers with them, right? And like, because that's something I did a lot in the summer, or when working on this project. I read a paper of some people. I had a bunch of questions about them, and then while previously maybe I would linger on those questions and fret about them and not find out the solution, there I really learned that most of the times researchers like talking about their papers, and you can just reach out to them, ask them if they have time to discuss their paper, and oftentimes when you discuss their paper with them, they're super happy that someone's interested in their work. So yeah, I learned not to be afraid to ask people to if they want to discuss their paper with me. What else
did you start Hans? If everyone, Hans is a really great reading group. If you're a practitioner, you should partake in it. Did you start the reading group around this time, or maybe?
Yes, exactly.
Okay, cool.
I had this experience that authors of papers are often very, very excited to talk about their research and very happy to spend time on that. And yeah, then I was looking for a reading group on graphs or about graphs, because I knew some reading groups about learning theory. I knew a reading group about reinforcement learning, but yeah, that were online, right? Of course, there are some graph reading groups at some university that the people at the university can go to, but I wanted a reading group that I can go to as well. That's just online and a Zoom meeting, and we can discuss a paper there. So after a few months of not finding the reading group that I was looking for, I started it myself, I suppose.
Cool. Okay. Congrats on that. Yeah, and maybe sorry for the sidetrack, but yeah, if you work on graphs, you should look into Hans' reading group. We'll post it in the description. It's really cool. Any other lessons for InfoMax, or maybe we can go to EquiBind.
Let's go to EquiBind.
Cool. You just have your names, your papers. They're like in America. There's various credit bureaus that do credit scores, and so there's Equifax. There's like various names. There's like names of companies that like do credit scores, and so your papers are kind of like credit bureau names. Okay. So it's what EquiBind. And so EquiBind was the paper you did at MIT. And really the breakthrough was really focused on ligand binding of targets. So you described small intrinsic properties of small molecules. EquiBind was more about predicting when small molecules interact with proteins.
Exactly.
And the key breakthrough is around sampling, which I still don't understand at all. So like I don't have any insight on this paper because I've read it twice and I still don't get it. But maybe you could talk about EquiBind, the story, and then kind of at least set up the paper, and then we could discuss it.
Yeah, so the task that we tackle in EquiBind, right, is predicting the structure in which a small molecule binds to a protein, right? And this is interesting for biologists or for drug discovery because sometimes if a small molecule binds to a particular spot of a protein, then the behavior of the protein is changed. Maybe some protein causes cancer, and if a small molecule binds to a particular location of that protein, then the abnormal or the cancer causing behavior of the protein is stopped. Yeah, and so this is a pretty helpful task for drug discovery if we can predict the location where a small molecule binds to a protein or attaches to a protein, right? And what we do in 3D InfoMax, or what previous approaches would always do, is they would try many different possible locations where the small molecule could go, and then score all of these locations or like evaluate them, and then use the best scoring location as their final prediction, right? But trying out all of these different positions takes a long time. And what we now do or what I think is the major takeaway of EquiBind is that this is also possible, or good 3D structure predictions are also possible in a single shot if you build in the prior knowledge about how 3D structures could interact in a good way. Yeah, and yeah, this new paradigm of predicting this in a single shot makes the method much faster, and this is very important for drug discovery, where we sometimes want to make predictions for millions of molecules when we search through a library of molecules to find one that has the behavior that we would like.
So when you mean single shot, so rather than scoring a bunch of locations on the target and then find the best one, single shot means you can find the best one as the first result based on all the data you have, or maybe that's not maybe that's too simplistic.
No, that's it. Based on the data that we train on, we have a single shot prediction, a single forward pass of our model that tells us tells us a 3D structure in which the small molecule probably binds to the protein. But of course, this isn't a solution to all our problems, right? I would really see this as it's not the perfect method. I would really see this as a demonstration. Okay, well, it has also some practical real world value, and actually some companies are using it in their pipelines, and they are happy with some of the results. But the main thing I see from the paper is a demonstration that it is possible to make these 3D structure predictions in a much faster way if we're using machine learning methods, or with these machine learning methods, and that this is something that we should also invest time in and look at, and not only into improving the score functions with which we evaluate the different locations.
I think it's kind of another theme of like sampling better, but also having better, maybe on the back end, having better data.
Yeah, but that's not what we're doing, right? We're not sampling better. We're not sampling at all.
Oh.
And that's this new paradigm, which we should also be looking at.
And so to not sample at all, what's the bar, at least for the protein structures you're working with, for the data? How many more different like configurations you need to map out for a given target to make single shot possible?
We train with the same type of data that the previous methods trained with, with which these scoring functions are also often trained with, because in the protein world, right, you just have data, or like you have all the data that's out there in these data banks, like the protein data bank PDB, with like I think around 180,000 protein structures, which are like the known protein structures as far as I know. And then we have PDB-bind, which is a subset of that, which are, as far as I can tell, all the known structures of small molecules bound to proteins. And yeah, we use those for training because that's all there is out there.
Oh, interesting. And so is there, like, I'm assuming there's some limitations in terms of some targets are not as well studied as others.
Mm-hmm.
And so a single shot is not going to be possible for maybe proteins or intrinsically disordered domains or something like that. There's always going to be a limitation in terms of our data.
Yeah, let's talk about limitations of EquiBind, right? Because there definitely are a big limitations, I think. So first of all, I believe to make a good 3D structure prediction, what we really want is to have some iterative approach, right? That reasons about the distances between the small molecule and the protein as the small molecules maybe fit into the pocket, or where our model thinks it will end up. Also, we know that, right, this whole thing is not in every case it is completely 100% determined where the small molecule will bind up. It is a stochastic process, and it is it's a probability distribution of where our small molecule is. And if we're able to model that would be maybe much better than having this single prediction. So if we have a generative model over where the small molecule could end up, this would be super valuable and maybe much more helpful in practice, and also reflecting reality much better. Yeah, and another thing is that we are not able to deal with symmetric proteins, right? Because currently we are always, even our training data, we have a symmetric protein where the small molecule is bound on one side of the protein. And our model predicts the small molecule now to be on the other side, which would be a just as good prediction. We will still tell the model it was wrong because in the training data it was on the other side of the symmetric protein. And yeah, if you have it would be great to have a generative model not necessarily a generative model, but for example, and that is able to tell us, yeah, with 50% probability it will land over here, and with 50% probability it will bind over there. That would be a much better model of what's going on in reality.
Cool. Yeah, I think every new problem, every limitation is an opportunity. Yeah. So maybe we can transition towards what you see long term in the field of like machine learning, molecular dynamics, and more. You've worked on InfoMax, EquiBind.
Yeah.
What's on the horizon for you? What other projects
Yeah.
you're exploring? Yeah, what's getting you excited?
Yeah, that's also something. Right, I've been thinking about that a lot, and also because I've been asked that a few times before. I've been thinking about this question, and I mean, keep in mind that I'm a two months in PhD student, and I'm not a
That's what makes you exceptional, Hans. You're 2 months in. You look at how productive you've been. So,
okay, thanks. But still, I'm not like David Baker or Max Welling or who have tons of experience and whose statements will be much more valuable about predicting the future, so to say, if you want to go with that. But yeah, maybe I'll just say what I'm really excited about. And that is maybe in this direction of, right, we've spent a lot of time modeling individual molecules and coming up with better graph neural networks, for example, to process molecules and making prediction about a single molecule. But what I think is really exciting is to model molecular interactions and the dynamic nature of molecules while they are interacting, which is of course core to the interactions of in many cases. And why am I so excited about molecular interactions and the dynamics of molecules there? Well, that's because, first of all, that is where a lot of impact is possible if we're able to model them, because it's what's important in drug discovery. And right in the end, in applications, we're not only interested in what a molecule looks like, so to say, but we're interested in what it does, so how it interacts with other molecules. Yeah, and the other thing, why the technical aspect of why I find this very exciting is because when modeling interactions, you run into even more interesting problems and mathematical hurdles to solve than when only modeling a single molecule. Because now you, for example, have two molecules floating around in space, and maybe you want to be equivariant with respect to the initial placement of one of those molecules. Yeah, and these symmetries that you can capture, they become a lot more intricate and even more interesting.
Cool. Yeah, I think and modeling small molecule protein interactions and protein-protein interactions are mind-boggling if you really think about it in the context of a cell.
It does not only have to be protein or small molecule protein, which we maybe often have in the drug discovery world. It can also be how a catalyst interacts with some material. If we're looking at there's this nice OC20 dataset from the Open Catalyst dataset. That's where we were trying to predict the dynamics of a small molecule interacting or coming close to some material. Yeah, and there are many different possible types of molecular interactions than just the ones in the drug discovery world, so to say. Yeah,
I'm a little biased, but I think totally agree, and I think you'd get along with Caddy Sar. I'll I'll email her. She's gonna love you. You'll get along really well with her. But yeah, any final thoughts? But this has been really great. I think great to have this initial conversation, and I know for a fact you're gonna do more work. Yeah, it's gonna be cool. You're gonna continue to publish more research. I'm gonna continue. I think we're all gonna read it. I think you're only 2 months in, so it'd be great to have a conversation maybe 2 years from now, and you'll be like, yeah, I have like 20 ICML paper. I'm just, you'll be a professor by then. Maybe you'll be a professor in 2 years. So
well, I hope maybe I would even say that I've done something wrong if I have 20 ICML papers by then, because yeah, I hope I produce impactful papers, and maybe not that many, but really important papers. And that is also something, yeah, maybe that is a good point to finish with, because that's something that my supervisors always tell me, right? Tommi, Jaakkola, and Regina, Barzilay, but also Octavian, which is to work on impactful or big and difficult problems, instead of picking out a thought or an idea that one has, which can maybe lead to the next ICML paper. Of course, if that is one's goal, or like if one just does research for the sake of curiosity, that is absolutely fine, right? That's many people do that just because you're curious. You work on a problem, and you just are playful and explore the world of research out of curiosity. And oftentimes this also produces super impactful work, right? But yeah, for me, this is only, or what means only, the curiosity and the fun of playing around is a huge part of why I love doing this research. But the other part that's important to me in the research is having impact and solving important and hard problems. And yeah, that is something that my supervisors and Octavian also always gave me as advice, is to find problems that not everyone could have solved, and to work on problems that are actually important to solve.
Cool. I think that's a great way to end it. Hans, thank you for taking the time to do this. I'm gonna press stop record right now, and then we can chat up later. I really appreciate you taking the time for this.
Likewise.